Hello everyone, welcome to Lab 11, Genetics. The study of genetics involves learning how traits are passed from one generation to the next. The science of genetics began with Gregor Mendel and his study of garden peas. Although the science of genetics began by studying plants, the concepts Mendel developed as a result of his experiments can also be used to explain many genetic traits in humans. In this week's lab, you'll learn about the laws Mendel formulated from his research and learn how these laws apply to the science of human genetics. To begin, let's review some important terms. Remember that housed within the nucleus of each of the cells in the human body is DNA. DNA, which stands for deoxyribonucleic acid, is our body's genetic material, the molecules that contain the information that make us who we are. The DNA in your cells is arranged into dark staining structures called chromosomes, and each chromosome consists of a single DNA double helix associated with special proteins called histones. Humans have 23 pairs of chromosomes, or 46 total chromosomes in each cell. 22 of those 23 pairs are referred to as autosomes, while the last pair includes the sex chromosomes, which determine whether you're male or female. Modern science has made it possible for us to view each of the chromosomes found in a human cell. Take a moment to look at the chromosomes in this figure. This set of human chromosomes is called a karyotype. As you can see, each human chromosome shown in the karyotype has a unique size, shape, and staining pattern. You should also have noticed that the first 22 pairs of chromosomes, the autosomes, are arranged in order of decreasing size and numbered accordingly. So chromosome pair 1 is the longest, and chromosome pair 22 is the shortest. The last pair of chromosomes, which we defined as the sex chromosomes, are not numbered. Instead, we simply refer to them as the sex chromosomes. In this particular figure, like the one in your notes, you're viewing a karyotype of a human male. How do you know that this person is male? Well, remember that genetically females have 2 X chromosomes and males have 1 X and 1 Y chromosome. And in this figure, you can see that clearly there is 1 X and 1 Y chromosome. Located along each of the chromosomes depicted in the karyotype at specific places called loci are genes, which control specific traits. So, for example, your eye color, hair texture, height, and blood type are all traits determined by your genes. Although we have 2 copies of each gene, both of which contain the same information, the specific base sequence in those genes may not be identical. Genes with different base sequences that code for the same trait are called alleles. Most of the genes in your genome have multiple alleles. For example, let's look at the pair of homologous chromosomes in this figure. Let's assume that the pair of homologous chromosomes in this figure is, for example, chromosome pair number 4. Notice that on each chromosome there are 3 genes highlighted. Gene 1 on both chromosomes is color-coded blue, indicating that these genes have the exact same DNA base sequence. Genes 2 and 3, however, are drawn in different colors on each chromosome, indicating that these genes have slightly different base sequences than their counterpart on the other chromosome. Although these genes are slightly different, they still code for the same trait. So, for example, if gene 2 is the gene that determines your hair color, you have 2 different versions of that gene. So you may have one gene that codes for blonde hair and one that codes for the production of brown hair. But again, although the genes code for different hair colors, they still determine the same overall trait, which in this case is hair color. Not all alleles are equal. You should recall from the lab on blood typing that some alleles are dominant and others are recessive. A dominant gene is one that hides or masks the presence of a second recessive allele, and a recessive allele is one whose presence is masked or hidden by a dominant allele. For example, the gene for dimples is dominant to the gene for lack of dimples. The dominant gene is written with a capital letter, and the recessive gene is written with a lowercase letter. Let's assume that gene 3 is the gene that determines whether you have or lack dimples. Okay, this gene from your mom is the recessive gene, while this gene from your dad is the dominant gene. This combination of genes is known as your genotype and should be written big D, little d. The physical trait that results from this combination of genes is called the phenotype, and in this case, the phenotype that results is dimples. Because we have a dominant gene in combination with the recessive gene is hidden, and so the physical trait that we see as a result is the presence of dimples. Continuing with the example of dimples, pause the podcast and take a moment to answer the next two questions. If you have dimples, there are two possible genotypes. You can either have two copies of the dominant gene or one dominant gene and one recessive gene. If you do not have dimples, your genotype must consist of two copies of the recessive gene. If you have two copies of the same allele, we say that you are homozygous for that trait. So, for example, if you have two copies of the dominant allele for dimples, you are homozygous dominant for that trait. If you have two copies of the recessive allele, you are homozygous recessive for that trait. If, however, you have one dominant allele and one recessive allele, you are heterozygous for that trait. As I mentioned earlier, Gregor Mendel established several important principles that we can use to explain how traits are inherited in humans. The first of his laws of inheritance is the principle of segregation. The principle of segregation says that the two alleles of a homologous pair of chromosomes separate during gamete formation such that each gamete receives only one allele. To better explain this principle, take a look at the screen. The process of meiosis results in the production of either 4 sperm cells or 4 egg cells. In this example, let's assume that the end product is 4 egg cells. Before those eggs are made, however, we have to make copies of each of the chromosomes in the cell. So to begin, the two chromosomes and the homologous pair are pulled apart and replicated. Once each chromosome has been copied, they will be separated and partitioned into one of the 4 egg cells that's produced. So to recap, the principle of segregation simply says that each of the chromosomes in a homologous pair will be separated and put into a different gamete as a result of meiosis. The principle of independent assortment is a little more complicated. It says that alleles of one gene pair assort independently of other gene pairs on non-homologous chromosomes. To better explain this, take a look at the screen. Notice here that there are 2 pairs of homologous chromosomes which have already undergone the process of replication. So at this point, each chromosome is ready to be partitioned into one of the 4 egg cells. So for example, the first chromosome may be partitioned into this egg cell, while this chromosome may be partitioned into this egg cell. But the important thing to remember here is that where the first chromosome ends up does not affect or determine where the other chromosome ends up. So for example, the 2 chromosomes may have been partitioned into the same cell, or the first chromosome may have been partitioned into a different egg cell. The main concept to understand with the law of independent assortment is that the way in which the chromosomes of one homologous pair are separated does not affect the way in which chromosomes of another homologous pair are separated, because these two processes are independent of each other. To predict the results of various genetic crosses, you must use Punnett squares, and you should remember Punnett squares from the lab on blood typing. To review, let's return to the example of dimples and work through the first question together. Okay, the question says, determine the genotypic and phenotypic ratios of offspring produced between a female who is homozygous dominant and a male who is homozygous recessive. Remember, to begin, you need to write the genotypes of both parents along the square. So the mother is homozygous dominant for dimples and has the genotype big D, which we'll write along the side of the square. The father is homozygous recessive, so along the top of the square we'll write little d, little d. To fill in the blanks in the square, simply cross-multiply across the block. So we have first big D times little d, giving us big D, little d. Take a moment to complete the remainder of the square. You should have noticed that the result is the same for each block. So there's a 100% chance that the children produced will have the genotype big D, little d, and a 100% chance that each child will have dimples. Each of the offspring produced in the genetic cross, again, is heterozygous for the trait of dimples. Each child is referred to as the first filial generation, or the F1 generation for short. Before moving on, pause the podcast and answer the next question in your handout. Another important concept in genetics is something called the product rule, which is used to predict the outcomes of multiple pregnancies. The product rule says that the probability of an event occurring is the product of the probabilities of each independent event. Let's work through the first question together. Okay, question 1 says, what is the probability of a heterozygous couple having 3 children with no dimples? Having completed the second Punnett square in the previous exercise, you should have determined the probability of a heterozygous couple having 1 child with no dimples is 1 out of 4, or 25%. But the question here wants to know the probability of having 3 children with no dimples. Okay, remember in math the term product means that we have to multiply something. So to find the answer, we have to multiply the probability of each independent event. So in this case we have 1/4 times 1/4 times 1/4, which gives us 1/64, which is about 1.6%. Pause the podcast and take a moment to answer question 2. Question 2 is set up exactly like the first question. Okay, first you have to know the probability of a heterozygous couple having 1 child who has dimples, and you should have determined a probability of 3 out of 4 from the Punnett square in the previous exercise. So because we want to know the probability of this couple having 4 kids with dimples, you should have said that mathematically we should have 3/4 times 3/4 times 3/4 times 3/4, which equals 81/256, or roughly 31.6%. As you can see, if you know the genotypes of both parents, you can easily determine possible genotypes and phenotypes of each child and the probabilities of specific traits that may result. Before continuing with the podcast, complete the next exercise in your handout, Mendelian Inheritance in Humans. At this point, we have made a few major assumptions in our discussion of the pattern of inheritance. We've assumed that each trait is completely controlled by 1 gene, that there are only 2 possible alleles for each gene, and that 1 allele is completely dominant to the other. However, human genetics can be a little more complicated. For example, in incomplete dominance, the phenotype of a heterozygous individual is intermediate between the phenotypes of homozygotes. A good example of incomplete dominance in humans is hair texture. Individuals who have 1 gene for curly hair and 1 gene for straight hair have hair that is intermediate between these two extremes. So in other words, they have wavy hair. In addition to incomplete dominance, codominance is also possible, in which two dominant alleles are simultaneously expressed. Remember that with human blood types, the A and B alleles are both dominant, and if you have type AB blood, you have both alleles which are both expressed. Another factor to consider when understanding human genetics is that there can be multiple alleles for a single gene. For example, there are 3 different genes for blood type and many more genes that determine height and eye color. However, it's important to remember that although there are multiple alleles for a gene within the human population, each individual person can have at most 2 alleles for that gene because our chromosomes are arranged in pairs. 2 additional terms that you should know are pleiotropy and polygenic. Pleiotropy means that there is one gene that determines many traits. A good example is a gene called SRY, which stands for sex-determining region of the Y chromosome. When activated, the SRY gene triggers the expression of many other genes which control the development of male sexual characteristics, such as the development of penis and testes. The term polygenic means that there are many genes that determine one trait. For example, human skin color is controlled by multiple genes. The last topic we need to discuss is called non-disjunction, which is the failure of chromosomes to separate completely during meiosis. The result of non-disjunction is sperm or egg cells with an abnormal number of chromosomes. If these sperm or eggs later combine with normal gametes, offspring may be produced with certain genetic abnormalities. That concludes this week's podcast. To complete this week's lab, follow the instructions for completing the problem set.